Good morning. Welcome to the spermatology laboratory at the University of the Western Cape. This morning we're going to introduce you to a new viability test. It's the nigrosine-eosine test. We combine the stains in a new way, and so this is a very important test to distinguish between a live and dead sperm. But in addition, this has now been automated by Microptic, so that the computer will decide which ones are pink and which ones are white. And of course, we can do it at a very low magnification, like 20 times, and therefore do many cells in a very short period of time. This morning I have with me master student Shannon Kaiser, who is going to assist me, and we will now explain to you how this technique works. Perhaps just before we start, I also need to mention that apart from the sperm functional test of live dead, the test also measures hypoosmotic swelling. We will now show you how the technique, how it is performed. So Shannon will now take the new nigrosine-eosine mix of Microptic. She will take 40 microliters, insert it in a amber Eppendorf tube. Yes, fill another one for the control, or another semen sample. So all of that is heated up to 37 degrees. And then the semen, 10 microliters of raw semen is taken, inserted in both tubes, and now she will take and carefully just mix the semen stain mixture. There's no need for vortexing. Vortexing will in any way damage the sperm. And we leave it now for 5 to 7 minutes before we will make the stain. Now we proceed to make the actual smear. And we use quite an ingenious technique. This technique was actually shown to me by my good friend and colleague, Dr. David Mortimer. And Shannon will show you how to make this. She takes 20 microliters of the mix that's been staining for approximately 5 to 7 minutes. She puts it in the center of the slide. And please notice how well this actually works. We now put the one slide on top of the other one, and the stain now spreads between the two slides. And once it has spread sufficiently, like she's showing there, she's going to actually pull the two slides away from each other. That is okay, I think. And what is nice about it is now we have two smears, and you can clearly see how nice the stain has come out. And actually we have duplicate slides, as you can see. I would have preferred it to be a bit darker. Here are examples of the correct darkness. You can see against the white, and it's really nice and even. I just need to make a note of caution here. While a lot of people would prefer to do it in semen, often semen is very viscous, and then it really needs to be washed. Because if you don't wash it in that instance, there will be a lot of what I would call background noise. Background noise meaning we will have all kinds of particles in the background. We won't have even distribution of the stain, and it really does not work well. It also produces kind of a network at the background, and so we advise very strongly that in the case of high viscous samples, particularly hyperviscosity, first wash the samples in a good medium. That would not affect the ratio of live to dead. Once the slides have dried completely, and this is really important, one must preferably wait overnight, and once they're completely dry, we will use a mounting medium such as DPX, the one that we prefer to use, and Shannon will now show you how to properly mount this, the slides which have dried. She puts 2 drops on, one on the one side, one on the other side, not too small, not too big, and that is well placed. And now she's going to mount it with 22mm by 50mm cover slips. These cover slips, please note, must be 0.17mm thick, because that would be the standard for the microscope to have the correct numerical aperture related to the thickness of the cover slip. So there it is, nicely mounted. And the moment it has spread, there's no need to wait for it to totally dry, because we're going to evaluate these slides, and this is one of the big advantages of this new technique, with by using only the ×20 objective. And the software has been developed by Microptic in such a way that it can very, very accurately distinguish between dark pink, light pink, and of course the white sperm, and accordingly between dead and live sperm. We will show that to you subsequently. After we have mounted the nigrosine using smear with DPX mounting medium, we can now study it. And we use brightfield microscopy. We use a 20 times objective, and this is the strength of this automated technique called BrightVit, that we use not very expensive components. It's plain brightfield microscopy, and also we can do many cells at a time because we only work with a 20 times objective. So firstly, I need to show you that here is the ACE 1300 new camera that is used. It has a very high resolution, and this is the camera of choice to be used for this purpose. We will now first of all change our module. I'm now going to demonstrate the software for Vitality, but this is a new feature of SCA Evolution, that one can select human bright field, the advantage that you just need bright field optics and only a 20 times objective. We enter the program, and I'm not going to show you all the details under configuration that has already been set up for this analysis. I'm going to press to go to the capture and analyze box. We can now look at nigrosine use in stained sections. We can focus sharply on these sperm, and we can start analyzing these sperm. But I'm going to show you areas that have very dark backgrounds versus very light backgrounds, and all the problems that may be associated, for example, this background noise here. But let us first start off with an import of the quality control file. And there you can clearly see now, if we go back to the results box, that the SCA system has detected correctly the white sperm, or the white sperm, compared to the single pink one. While we are at this point, it's important to show you that this test has not only been designed as a sperm functional test for vitality, in other words distinguishing between alive and dead, but also as a hypoosmotic swelling test. And it's interesting in this instance that 3 of the 4 white sperm, live sperm, have shown perfect osmotic swelling. You can see there the tail is folded back, and usually there's a huge droplet sitting there. Similarly here,
whereas the dead sperm has straight tails, and so the membrane burst. One can go back to the results section. There you can summarize the results, and of course you can create a report. And here you can see again, very useful, a pie diagram, a little table, and the actual proof of the pudding that it indeed detected it correctly. And this is very important in terms of quality control, to ensure that each time your instrumentation is set up to detect correctly. We will now show live capture, and you can see here we have an example where we need to focus really well. Look at the nice dark background, and let's see how the analysis goes. And quite well analyzed. If we go back to that particular analysis, which is right at the end here, and view that field, you will see that sperm have been extremely well captured, particularly those 1, 2, 3, 4, 5 pink cells. Most of the white cells have been captured, but of course when they overlap it won't work. But then if you really want to add these, because they may make a difference, you can add them really very, very quickly. Add live, add live, add live. And so even if there are mistakes, or tiny mistakes, it can be very, very easily rectified. So you can see that we have quite different backgrounds on a stain of this nature. If I move it around, can you see there are areas of real big brightness, and avoid those. Or if you cannot, turn your light down as much as you can and then try further analysis. For example, in this instance, we would probably get away with this background, finely focused, and analyze. View the field, and so we've captured some 13-14 fields of this sample, and by moving this cursor we can go from field to field, and you can see by and large most sperm have been captured, even when the pink is very faint. Where there are small mistakes, we can rectify it so that it's 100% correct, and this is great that we have control over the software in the end. So this brings us to the end of this demonstration of the nigrosine eosine hyperosmotic swelling test, both how you perform it and also how the software of Microptic helps to actually evaluate the different types. I want to thank Shannon Kaiser for helping me with this, and we hope you've enjoyed it.